Ramachandran plot. CSIR-NET JRF Life Sciences Ultra Detailed Lecture. Paragraph format. No tables. Maximum word limit. Tailored to crack the exam. Includes PYQs with year and answers. The Ramachandran plot, introduced by G. N. Ramachandran and colleagues in the early 1960s, is a graphical representation of the conformational angles that are permissible for amino acid residues in a polypeptide chain. It is one of the most foundational tools in structural biology, particularly for understanding protein folding, secondary structure formation, and stereochemical constraints on peptide backbone conformations. In the CSIR-NET Life Sciences exam, questions often test your understanding of allowed and disallowed regions, the conformational flexibility of amino acids, and its implications for protein structure prediction and validation. 1. Basis of the Ramachandran plot. Proteins are composed of amino acids linked by peptide bonds, which are planar and rigid due to partial double bond character. However, rotation is possible around the two single bonds flanking the peptide bond. These are phi the angle of rotation around the NC alpha bond. Psi the angle of rotation around the C alpha C cardinal bond. Because the peptide bond is planar, the only degrees of conformational freedom in a protein backbone come from phi and psi. However, not all combinations of phi and psi are sterically allowed due to van der Waals repulsion between atoms. The Ramachandran plot maps these angles on the x-axis phi and y-axis psi, indicating which combinations are sterically favorable, allowed, and which are disallowed due to steric clashes. 2. Features of the Ramachandran plot. The plot is a square graph with phi values typically ranging from -180 degrees to +180 degrees along the x-axis and psi values also from -180 degrees to +180 degrees on the y-axis. Within this space, only certain regions are allowed for most residues due to steric hindrance. Allowed regions. The upper left quadrant phi approximately equals -60 degrees, psi approximately equals -45 degrees to -60 degrees corresponds to beta sheet conformations. The lower left quadrant phi approximately equals -60 degrees, psi approximately equals -40 degrees corresponds to the right-handed alpha helix. The upper right quadrant phi approximately equals +60 degrees, psi approximately equals +60 degrees is where left-handed alpha helices may occur, though they are rare. Disallowed regions. These areas have phi-psi combinations that result in severe steric hindrance and are thus rarely occupied except by allowed regions. The upper left quadrant phi approximately equals -60 degrees, psi approximately equals -45 degrees to -60 degrees corresponds to beta sheet conformations. The lower left quadrant phi approximately equals -60 degrees, psi approximately equals -40 degrees corresponds to the right-handed alpha helix. The upper right quadrant phi approximately equals +60 degrees, psi approximately equals +60 degrees is where left-handed alpha helices may occur, though they are rare. Disallowed regions. These areas have phi-psi combinations that result in severe steric hindrance and are thus rarely occupied except by specific residues under constrained conditions. Glycine. Because it lacks a side chain, its R group is a single hydrogen atom. Glycine has greater conformational flexibility and can occupy regions forbidden to other amino acids. Proline. Due to its cyclic structure and rigid pyrrolidine ring, proline is highly restricted in its phi angle and thus occupies a narrow region in the plot. The Ramachandran plot serves as a quality control tool in structural biology. High-resolution X-ray and NMR structures are evaluated based on how many residues lie within allowed regions. Deviations may indicate errors in structure determination. 3. Applications in biology and CSIR-NET related concepts. Secondary structure identification. The phi and psi angles associated with alpha helices and beta sheets fall within distinct regions of the plot, making it possible to predict or confirm secondary structure content in a protein. Protein structure validation. Most residues in a properly folded protein should fall within allowed regions. Modern bioinformatics tools use the Ramachandran plot to assess the stereochemical quality of crystallographic protein models. Molecular dynamics and folding simulations. Simulations of protein dynamics use phi-psi conformational maps to explore energetically feasible conformations during folding or docking. Understanding unusual conformations. Deviations in the plot can indicate functionally important regions such as turns, loops, or catalytic residues under structural strain. 4. Previous year CSIR-NET questions. Question 1. CSIR-NET Deck 2016. Which of the following amino acids has the most conformational flexibility and can populate all four quadrants of the Ramachandran plot? A. Glycine. B. Proline. C. Valine. D. Cysteine. Answer. A. Glycine. Explanation. Glycine, with its minimal side chain, experiences the least steric hindrance and can adopt a wide range of phi and psi angles. Question 2. CSIR-NET December 2015. The Ramachandran plot for a newly determined protein structure shows many residues lying in disallowed regions. What does this likely indicate? A. Protein has many proline residues. B. Protein is composed entirely of glycine. C. Possible errors in structure determination. D. Protein is membrane bound. Answer. C. Possible errors in structure determination. Explanation. A high number of residues in disallowed regions suggests structural artifacts, not a biologically realistic model. Question 3. CSIR-NET June 2019. Which of the following statements is false regarding the Ramachandran plot? A. Proline shows limited phi angles due to its rigid ring structure. B. Glycine has restricted conformations due to steric hindrance. C. Right-handed alpha helix occupies the lower left region of the plot. D. Most residues fall in specific allowed regions based on secondary structure. Answer. B. Glycine has restricted conformations due to steric hindrance. Explanation. This statement is false. Glycine actually has greater conformational freedom due to the absence of a bulky side chain. Glycine actually has greater conformational freedom due to the absence of a bulky side chain. 5. Summary and final notes. The Ramachandran plot is a powerful theoretical and practical framework to understand protein backbone flexibility and constraints. In the context of CSIR-NET, expect questions that test your ability to interpret the plot, identify structural regions, correlate amino acid properties with conformational freedom, and evaluate protein model quality. It also integrates knowledge from protein chemistry, structural biology, and bioinformatics, making it a highly interdisciplinary topic. Mastery of this concept is essential for understanding protein folding, enzyme active site geometry, and molecular evolution. Let me know when you're ready for the next topic. I'll continue in the same detailed format. You said. Secondary structure. ChatGPT said. Secondary structure of protein. CSIR-NET JRF Life Sciences Ultra Detailed Lecture. Maximum detail. No tables. Paragraph only. Tailored to crack the exam. Includes 2 to 3 PYQs with year and answers. The secondary structure of proteins refers to the regular, repeating conformations of the polypeptide backbone stabilized primarily by hydrogen bonds. These structures arise due to the rotation of the phi and psi angles of the peptide backbone, which are constrained by steric hindrance as depicted in the Ramachandran plot. The most common and significant secondary structural elements in proteins are the alpha helix, beta pleated sheet, beta turn, and loops or coils. These structures provide the first level of organized three-dimensional architecture in proteins and form the scaffold for tertiary structure development. Their stability and geometry are governed by hydrogen bonding patterns, residue type, and backbone flexibility. 1. Alpha helix. The alpha helix is a right-handed coiled structure stabilized by intrachain hydrogen bonds between the carbonyl oxygen C equals O of the ITH residue and the amide hydrogen NH of the I plus fourth residue. Each turn of the helix contains approximately 3.6 amino acid residues, and the rise per residue is about 1.5 A, resulting in a pitch height of 1 complete turn of 5.4 A. This regular H bonding pattern makes the alpha helix highly stable. The side chains project outward from the helical backbone, minimizing steric hindrance. The alpha helix is prevalent in globular proteins and transmembrane domains, especially in hydrophobic segments. Not all residues favor helix formation. Proline, due to its rigid ring, disrupts alpha helices and is called a helix breaker, while glycine, due to its conformational flexibility, also tends to disrupt the helix. In contrast, alanine is highly helix promoting. The N-terminal of the alpha helix often carries a partial positive charge, and the C-terminal carries a partial negative charge due to the alignment of dipoles from multiple peptide bonds. 2. Beta sheet, beta pleated sheet. The beta sheet. Each turn of the helix contains approximately 3.6 amino acid residues, and the rise for residue. The secondary structure of proteins refers to the regular, repeating conformations of the polypeptide backbone stabilized primarily by hydrogen bonds. These structures arise due to the rotation of the phi and side angles of the peptide backbone, which are constrained by steric hindrance as depicted in the Ramachandran plot. The most common and significant secondary structural elements in proteins are the alpha helix, beta pleated sheet, beta turn, and loops or coils. These structures provide the first level of organized three-dimensional architecture in proteins and form the scaffold for tertiary structure development. Their stability and geometry are governed by hydrogen bonding patterns, residue type, and backbone flexibility. 1. Alpha helix. The alpha helix is a right-handed coiled structure stabilized by interacting hydrogen bonds between the cardinal oxygen C equals O of the ITH residue and the amide hydrogen NH of the I plus fourth residue. Each turn of the helix contains approximately 3.6 amino acid residues, and the rise per residue is about 1.5 A, resulting in a pitch height of one complete turn of 5.4 A. This regular H bonding pattern makes the alpha helix highly stable. The side chains project outward from the helical backbone, minimizing steric hindrance. The alpha helix is prevalent in globular proteins and transmembrane domains, especially in hydrophobic segments. Not all residues favor helix formation. Proline, due to its rigid ring, disrupts alpha helices and is called a helix breaker, while glycine, due to its conformational flexibility, also tends to disrupt the helix. In contrast, alanine is highly helix promoting. The N-terminal of the alpha helix often carries a partial positive charge, and the C-terminal carries a partial negative charge due to the alignment of dipoles from multiple peptide bonds. 2. Beta sheet, beta pleated sheet. The beta sheet structure consists of extended beta strands linked together by interstrand hydrogen bonds. These sheets can be parallel strands run in the same direction, or anti-parallel strands run in opposite directions, with anti-parallel sheets generally being more stable due to more linear H bonds. The R groups of adjacent residues alternate above and below the plane of the sheet, reducing steric clash. The distance between residues in a beta strand is approximately 3.5 A, which is greater than in alpha helices, giving beta sheets a stretched appearance. Amino acids like valine, isoleucine, and phenylalanine are often found in beta sheets due to their branched or bulky side chains. In beta barrel proteins, beta sheets are arranged in a cylindrical manner, forming pores or channels across membranes, such as in porins. 3. Beta turns and loops. Beta turns, or reverse turns, are tight loops that reverse the direction of the polypeptide chain, often connecting the ends of two adjacent strands of an anti-parallel beta sheet. A typical beta turn involves four residues, and the cardinal oxygen of residue I hydrogen bonds with the amide hydrogen of residue I plus 3. These turns are stabilized by internal hydrogen bonding and are crucial for compact folding. Glycine due to flexibility and proline due to kinks are commonly found in beta turns. Loops are irregular regions that do not have a regular pattern of H bonding. They are flexible and often found on protein surfaces, serving as sites for ligand binding, enzyme activity, or recognition. Though they lack regularity, loops are functionally important and structurally conserved. 4. Stabilization and prediction. Secondary structures are stabilized by hydrogen bonding, dipole interactions, and side chain compatibility. The Ramachandran plot is an essential tool to identify low phi and psi angles for different secondary structures. Modern secondary structure prediction algorithms like Chokas and Arbor rely on residue preferences and sequence motifs to predict alpha helices, beta sheets, and turns. Experimental identification is typically done using X-ray crystallography, NMR spectroscopy, or circular dichroism CT spectroscopy, which can determine the proportion of alpha and beta content based on absorbance and magnetic behavior of peptide bonds. 5. Functional significance. Secondary structures are not only structural features but also functionally relevant. Alpha helices often form transmembrane domains, and their amphipathic nature, hydrophilic on one side and hydrophobic on the other, is critical in protein membrane interactions. Beta sheets contribute to the mechanical strength of proteins like silk fibrin and form the basis for amyloid fibrils in prion diseases. Turns and loops provide flexibility, specificity, and active site accessibility in enzymes. 6. Previous year questions from CSIRNet. Question 1. CSIRNet June 2013. In a protein structure, the alpha helix is primarily stabilized by A. Hydrophobic interactions. B. Hydrogen bonding between side chain. C. Ionic interactions. D. Hydrogen bonding between backbone atoms. Answer. D. Hydrogen bonding between backbone atoms. Explanation. The alpha helix is stabilized by intermolecular H bonds between the cardinal oxygen and amide hydrogen 4 residues apart in the backbone. Question 2. CSIRNet Deck 2017. Which amino acid is most likely to be found at the turn in a beta turn? A. Leucine. B. Isoleucine. C. Proline. D. Valine. Answer. C. Proline. Explanation. Due to its cyclic structure, proline induces a kink in the polypeptide chain and is frequently found in beta turns. Question 3. CSIRNet June 2015. In an antiparallel beta sheet, the hydrogen bonds between strands are A. Random and weak. B. Linear and strong. C. Bent and weak. D. Ionic in nature. Answer. B. Linear and strong. Explanation. Antiparallel beta sheets have linear H bonds, which are more stable than the bent H bonds in parallel beta sheets. 7. Conclusion. A deep understanding of secondary structure is crucial for mastering protein biochemistry and for success in CSIRNet JRF Life Sciences. The ability to distinguish between helices, sheets, and turns, understand the residues involved, their stabilization, and experimental validation methods will help answer both direct and application-based questions. Given the recurrent nature of these questions in past CSIR papers, this topic is a high-yield area and must be prepared thoroughly. Let me know when you're ready for the next topic. I'll continue in the same style. You said, domains. ChatGPT said, domains in proteins. CSIRNet JRF Life Sciences Ultra Detailed Theory Lecture. Paragraph format only. No tables. Maximum word limit. Previous year CSIRNet questions included with answers in here. In protein biochemistry, the term domain refers to a distinct, compact, and independently folding structural and functional unit within a polypeptide chain. Domains are often composed of 50 to 300 amino acids and can function independently of the rest of the protein, which distinguishes them from smaller motifs or secondary structural elements. Protein domains are often associated with specific biochemical functions, such as binding to DNA, catalysis, ligand recognition, or membrane insertion. These domains often retain their functional and structural properties even when isolated from the full-length protein, making them critical evolutionary and functional units. Domains arise during evolution as modular building blocks. Through gene duplication and recombination events, new proteins with multiple domains evolve to perform complex functions. Proteins that share the same domain often share a common evolutionary origin, which is reflected in their sequence and structural homology. This modular architecture allows proteins to perform multifunctional roles by combining different domains in a single polypeptide. For instance, a signaling protein may have a receptor binding domain, a transmembrane domain, and a catalytic kinase domain, each responsible for a specific role. Structurally, domains can be classified into several types, including all alpha domains, all beta domains, alpha beta domains with alternating alpha helices and beta sheets, and alpha plus beta domains with segregated alpha and beta structures. These conformations are stabilized by hydrophobic core interactions, hydrogen bonding, and electrostatic forces. Domains are identified by X-ray crystallography, NMR spectroscopy, or computational methods such as sequence alignment, domain prediction software, and fold recognition algorithms. Functionally, domains enable protein-protein interactions, catalytic activity, signal transduction, DNA, RNA binding, and membrane anchoring. A classic example is the SH2 domain SRC homology 2, which binds specifically to the phosphotyrosine-containing motifs in signaling proteins, and the zinc finger domain, which binds to DNA and regulates gene transcription. Another example is the immunoglobulin domain, found in antibodies and cell surface receptors, that forms a beta sandwich structure providing structural integrity. Domains also contribute to the evolutionary plasticity of proteins. Because domains can function and fold independently, they serve as evolutionary modules that can be rearranged and reused in different proteins. This modularity contributes to the diversity of the proteome and is a reason for the complexity of higher organisms despite relatively limited numbers of genes. From the perspective of bioinformatics, databases such as FAM, Interpro, and SCOP, structural classification of proteins catalog protein domains based on sequence similarity and structural folds. These resources help predict the function of unknown proteins by identifying conserved domains. In drug design, targeting specific domains of a multi-domain protein can enhance selectivity and reduce off-target effects, especially in enzymes and receptors. Misfolding or mutation in specific domains can result in loss of function or gain of toxic function, leading to diseases. For example, mutations in the DNA binding domain of p53 protein compromise its tumor suppressor activity and are implicated in cancer. Similarly, domain deletion or rearrangement can lead to genetic disorders due to loss of regulatory control in developmental proteins. In multi-domain proteins, domains are often connected by flexible linker regions that allow conformational rearrangements. These flexible junctions permit domain movements essential for allosteric regulation and substrate channeling. Enzymes like pyruvate dehydrogenase utilize multiple domains and subunits to carry out complex multi-step reactions within a confined structure, ensuring efficiency and specificity. Previous year CSIRNet questions on domains. Question 1. CSIRNet Deck 2016. Which of the following best defines a domain in a protein? A region composed entirely of beta sheets. B. A compact, independently folding unit with a specific function. C. A sequence motif repeated throughout the protein. D. A linear sequence between two cysteine residues. Answer. B. A compact, independently folding unit with a specific function. Explanation. Domains are structurally and functionally distinct regions of a protein that can fold independently. Question 2. CSIRNet June 2014. Which of the following statements about protein domains is not correct? A. Domains are structurally stable and functionally autonomous. B. Domains are always formed from continuous stretches of amino acids. C. Multiple domains can exist in a single protein. D. The same domain can be found in different proteins. Answer. B. Domains are always formed from continuous stretches of amino acids. Explanation. While many domains are formed from continuous stretches, some domains are composed of discontinuous sequences that fold together into a compact structure. Question 3. CSIRNet December 2015. A DNA binding protein is found to have a helix-turn helix motif. What is most likely true about this structure? A. It functions as a domain that recognizes phospholipids. B. It acts as a protease site. C. It contributes to the protein's DNA binding domain. D. It allows the protein to act as a chaperone. Answer. C. It contributes to the protein's DNA binding domain. Explanation. The helix-turn helix is a structural motif commonly found within DNA binding domains of transcription factors. Conclusion. Protein domains are critical for understanding structure-function relationships, evolution, and molecular mechanisms in biology. Their modular nature and ability to function independently have made them central to bioinformatics, structural biology, and therapeutic design. The presence of domain-related questions in multiple CSIRNet exams highlights their importance in the syllabus. A strong grasp of this topic enables students to answer questions related to protein structure, evolution, and function with accuracy. Mastery of domain architecture can also aid in tackling interdisciplinary questions that combine biochemistry, molecular biology, and structural bioinformatics. Let me know when you are ready for the next topic. I'll continue in the same depth and CSIR exam-specific style. You said, motif. ChatGPT said, motif in proteins. CSIRNet JRF Life Sciences full-length theory lecture based to advanced level, no tables, paragraph format only, highly detailed, includes previous CSIRNet questions with answers in years. In the study of proteins, a motif refers to a short, conserved sequence or structural pattern within a protein that is associated with a specific function or structural role. Unlike domains, motifs do not usually fold independently or function autonomously, but they contribute critically to the overall structure and function of proteins. Motifs can be sequence motifs, which are conserved linear sequences of amino acids, or structural motifs, which represent conserved 3D arrangements of secondary structures like alpha helices and beta strands. Sequence motifs are commonly identified through multiple sequence alignments in bioinformatics analyses. For instance, the N-glycosylation motif ASNXS or THR, where X does not equal proline, is a short, conserved sequence recognized by glycosylation enzymes in the endoplasmic reticulum. Another well-known example is the WACRA motif GXXXGKST, present in many ATP binding proteins, including kinases and helicases. Such motifs are critical for ATP recognition and binding. Structural motifs, on the other hand, are recognized by their 3D conformation rather than their linear amino acid sequence. An archetypal example is the helix-turn helix motif, commonly found in DNA binding proteins. It comprises two alpha helices connected by a short turn. One helix fits into the major groove of DNA, facilitating sequence-specific interaction. Similarly, the zinc finger motif involves a conserved arrangement of cysteines and histidines that coordinate a zinc ion, stabilizing a fold that can bind DNA, RNA, or other proteins. It is crucial to understand that motifs are not always functionally autonomous. Instead, they typically mediate a specific biochemical activity as part of a larger domain or protein. For instance, the P-loop motif is part of a domain involved in phosphate binding and hydrolysis, but the motif alone is insufficient for complete function without the rest of the domain. From an evolutionary perspective, motifs are highly conserved because small changes can disrupt crucial biochemical interactions, such as ligand binding or catalytic activity. This conservation allows motifs to be used as signatures or markers in bioinformatics to predict the function of unknown proteins. For example, the EF hand motif, a helix-loop helix structure involved in calcium binding, can be used to identify calcium binding proteins in genomic data. Motifs are also essential in signal transduction pathways, where they mediate specific interactions between proteins. For example, SH3 binding motifs, which typically have a PXXP consensus sequence, bind to SH3 domains in signaling proteins. This specificity ensures correct pathway activation and cellular response. Biologically, motif integrity is essential. Mutations in conserved motifs can lead to loss of function or gain of toxic function, resulting in disease. For instance, mutations in the WACRA motif of mitochondrial ATP synthase can impair ATP production, leading to mitochondrial disorders. In research, motifs are used in mutational analyses, protein engineering, and drug targeting. By modifying or mimicking motifs, researchers can alter protein function or inhibit pathological interactions. Drugs targeting motif-mediated protein-protein interactions are a growing area in therapeutics, particularly in cancer and neurodegeneration. Motifs are cataloged in databases such as ProCyte, FAM, and SMART, which help researchers predict protein function and evolutionary relationships. These databases are frequently updated with motif information derived from experimental and computational studies. To summarize, motifs are short, conserved elements critical for protein function and interaction, often working as part of larger structural or functional units like domains. Mastery of protein motifs is essential for understanding protein function, predicting activity from sequence data, and interpreting experimental results in molecular biology and biochemistry. Previous year CSIRNet questions on motifs. Question 1. CSIRNet Deck 2017. The P-loop motif in proteins is associated with A. Protein degradation. B. Phosphorylation of tyrosine residue. C. Binding to phosphate groups, such as in ATPD membrane insertion of proteins. Answer. C. Binding to phosphate groups, such as in ATP. Explanation. The P-loop or phosphate binding loop motif is commonly found in ATP and GTP binding proteins. Question 2. CSIRNet June 2013. Which of the following best differentiates a domain from a motif? A. Motif is always catalytic. Domain is always structural. B. Motif is a short conserved sequence. Domain is a larger independently folding unit. C. Domain is not functional, but motifs are deep. Motifs and domains are interchangeable terms. Answer. B. Motif is a short conserved sequence. Domain is a larger independently folding unit. Explanation. This is a textbook distinction. Motifs are shorter and do not fold independently, while domains do. Question 3. CSIRNet Deck 2018. Which of the following is a DNA binding motif? A. Helix turn helix. B. P-loop. C. SH2 domain. D. Transmembrane alpha helix. Answer. A helix turn helix. Explanation. Helix turn helix is a classic DNA binding structural motif found in many transcription factors. Conclusion. Motifs are short, essential building blocks in protein structure and function, often acting as functional hotspots within larger domains. Their conservation, simplicity, and specificity make them fundamental to understanding molecular recognition, catalysis, and regulation in biological systems. In the context of CSIRNet Life Sciences, understanding motifs provides a conceptual bridge between protein structure and function, and this topic often appears in both direct and concept-based questions. Ensure that you are familiar with examples like helix turn helix, P loop, zinc finger, and EF hand, as these are frequently tested. Let me know the next topic and I'll continue building your high-quality CSIR net coaching notes accordingly. You said, fold. You said, folds. ChatGPT said, folds in proteins. CSIR net JRF Life Sciences Ultra Detailed Theory Lecture. Base to advanced level paragraph format only, maximum word limit, previous year questions with answers in years. Protein folds refer to the three-dimensional arrangements of secondary structure elements, alpha helices, beta sheets, and loops that create the overall architecture of a protein or a domain. A fold is a stable, recurrent structural pattern that can be found across proteins with little or no sequence similarity. The concept of folds is central to structural biology, as it provides a framework to understand how proteins achieve stability, specificity, and function despite high sequence diversity. Folds are larger than motifs but smaller than entire proteins, often representing the structural core of a functional domain. Many folds are conserved across evolution, implying that structure is more conserved than sequence. For example, the Tim barrel fold, found in Triosophisphate isomerase, consists of alternating alpha helices and beta strands arranged in a barrel-like structure. This same fold is found in a wide range of enzymes, regardless of their catalytic function or amino acid sequence, underlying the principle of convergent evolution of structure. A fold often consists of a specific topology, the connectivity and arrangement of helices and strands, which is crucial for protein folding and function. The Rossmann fold, for instance, consists of a six-stranded parallel beta sheet flanked by alpha helices on both sides and is commonly seen in nucleotide binding proteins. The stability of such folds arises from hydrophobic core packing, hydrogen bonding, and van der Waals interactions, all of which are optimized in these recurrent structural frameworks. Structural bioinformatics has classified protein folds into hierarchical levels, class e.g., all alpha, all beta, alpha beta fold, superfamily, and family. These classifications are organized in databases like SCOP, Structural Classification of Proteins, and CAF, which help scientists predict function and evolutionary relationships. A single fold can support diverse functions. For instance, the immunoglobulin fold is used in antibodies, cell adhesion molecules, and receptors. During protein folding, these folds emerge through a series of intermediate states, often guided by chaperones and folding catalysts. Misfolding of these structures can result in pathological conditions, such as prion diseases and amyloidosis, highlighting the importance of correct fold formation. Folding funnels, an energy landscape model, describe how protein sample conformations en route to their native fold, driven by thermodynamic stability and kinetic accessibility. Importantly, while some folds are unique to specific protein families, others, like the beta propeller fold or alpha beta hydrolase fold, appear in multiple, unrelated protein groups, making them structural scaffolds reused by evolution. The modular nature of folds allows for domain recombination, producing multi-domain proteins with complex functions. From a CSIR net perspective, folds are a key topic that ties together protein structure, function, and evolution. Questions may ask for identification of fold types, structural features, or their functional significance. Understanding folds not only aids in functional annotation of proteins in genome projects but also in rational drug design, where inhibitors are built to fit specific structural folds of enzymes or receptors. Previous year CSIR net questions on protein folds. Question 1, CSIR net deck 2019. Which of the following protein folds is commonly associated with nucleotide binding proteins? A. Immunoglobulin fold. B. Zinc finger fold. C. Rossmann fold. D. Beta barrel fold. Answer: C. Rossmann fold. Explanation: the Rossmann fold is a structural motif involved in binding nucleotides, especially NADP plus cofactors. Question 2, CSIR net June 2016. Which of the following best describes the Tim barrel fold? A. Alternating alpha helices and beta strands forming a barrel. B. Only alpha helices forming a coil. C. Beta sheets with loops connecting them. D. Parallel beta sheets with external helices. Answer: alternating alpha helices and beta strands forming a barrel. Explanation: the Tim barrel fold consists of 8 alpha helices and 8 beta strands alternating in a barrel arrangement. Question 3, CSIR net deck 2014. Protein folds are evolutionarily conserved because A. Their sequences are always identical. B. Their 3D structures allow for versatile and stable function. C. They cannot mutate. D. Their RNA sequences code for multiple conformations. Answer: B. Their 3D structures allow for versatile and stable functions. Explanation: despite sequence variability, conserved folds are maintained due to their stability and functional significance. Conclusion: protein folds represent one of the most fundamental levels of biological organization in structural biology. They serve as conserved structural solutions to the problem of molecular stability and function. By understanding folds, one gains insight into the evolutionary plasticity of protein structures, the relationship between structure and function, and the principles guiding protein engineering and drug development. For CSIR net Life Sciences, mastering protein folds enhances your conceptual clarity in protein biochemistry and helps decode advanced structural biology questions. Always remember that while sequences may diverge, folds are remarkably conserved, making them central to modern biology. Let me know the next topic from your CSIR net syllabus, and I'll continue preparing your premium level coaching notes accordingly. You said. Conformation of nucleic acids. ChatGPT said. Confirmation of nucleic acids. CSIR net JRF Life Sciences premium level lecture notes. Maximum detail. Note tables. Paragraph format only. Based on previous year question areas. 2 to 3 PYQs with answers in years. Nucleic acids, primarily DNA and RNA, are long-chain polymers composed of nucleotides. The conformation of nucleic acids refers to the three-dimensional arrangement of their atoms in space. This spatial structure is not fixed. It is dynamic and influenced by base composition, ionic environment, hydration, and interaction with proteins or drugs. Conformation is essential because it determines the biological activity, replication fidelity, transcription, and interactions with enzymes. The primary structure of nucleic acids is a linear sequence of nucleotides linked by phosphodiester bonds. However, the secondary and tertiary conformations are formed by hydrogen bonding between bases and base stacking interactions, which stabilize the structure. DNA conformations: A, B, and Z forms. The most common and biologically relevant conformation of DNA is the bDNA, which was described by Watson and Crick. It is a right-handed helix with about 10.5 base pairs per turn and a diameter of approximately 20 A. The bases are perpendicular to the helical axis, and the sugar adopts the C2 feet endopucker. The major and minor grooves in bDNA are critical for sequence-specific binding of regulatory proteins. Under dehydrated conditions, aDNA is formed. It is also a right-handed helix but is shorter and wider than bDNA. It has about 11 base pairs per turn, and the bases are tilted relative to the helix axis. The sugar pucker changes to C3 feet endo. Though not common in cellular DNA, aDNA is found in DNA-RNA hybrids and double-stranded RNA. zDNA is a left-handed helical form of DNA that occurs in alternating purine pyrimidine sequences, like CGCGCG, under high-sulfur supercoiling stress. zDNA has a zigzag sugar phosphate backbone, with a diameter of approximately 18 A. It has 12 base pairs per turn. The biological significance of zDNA lies in its regulatory role, especially in transcriptional control and chromatin remodeling. RNA conformations. Unlike DNA, RNA is usually single-stranded but can fold into complex secondary and tertiary structures due to interstrand base pairing. Common secondary structures include hairpin loops, stem loops, bulges, and internal loops. These features contribute to RNA's functional diversity in translation, catalysis ribozymes, and regulation mRNA. The A-form helix is the predominant helical conformation adopted by double-stranded RNA, dsRNA. This makes RNA molecules inherently more rigid and less flexible than DNA. The 2' group of ribose in RNA not only promotes this conformation but also makes RNA more chemically labile. Tertiary RNA structures involve pseudoknots, kissing loops, and coaxial stacking, stabilized by Mg² ions and hydrogen bonding. Ribosomal RNA, rRNA, and transfer RNA turn up exhibit highly folded tertiary conformations essential for protein synthesis. The cloverleaf secondary structure of turn folds into an L-shaped tertiary structure, crucial for its interaction with ribosomes and amino acyl turn synthetases. Biological implications of nucleic acid conformation. The conformation of nucleic acids is essential for replication, transcription, translation, and DNA repair. Specific conformational forms are recognized by DNA-binding proteins, transcription factors, and nucleases. Changes in DNA conformation, such as B to Z transition, can act as signals for gene regulation. Supercoiling, another level of DNA conformation, plays a major role in chromosome compaction and gene expression. Enzymes like topoisomerases modulate supercoiling, and inhibitors of these enzymes are used as anti-cancer agents. Chromatin conformation in eukaryotes, regulated through nucleosome positioning and histone modifications, is also central to transcriptional control. Furthermore, chemical modifications like methylation can influence the conformation and hence the function of nucleic acids. Methylation of cytosine and CpG islands often leads to gene silencing and is associated with epigenetic regulation. In biotechnology and medicine, understanding nucleic acid conformation aids in the design of antisense oligonucleotides, RNA-based therapeutics, and CRISPR SAW systems. RNA's ability to form catalytically active structures supports the RNA world hypothesis, where RNA preceded both DNA and proteins in evolution. Previous year CSIRNet questions on nucleic acid conformation. Question 1. CSIRNet June 2019. Which of the following statements about cDNA is correct? A. It is right-handed and has 10.5 BP per turn. B. It is left-handed and found in alternating purine pyrimidine region. C. It is the most common conformation of cellular DNA. D. It lacks a helical structure. Answer. B. It is left-handed and found in alternating purine pyrimidine regions. Explanation. zDNA is a left-handed helix typically formed in CG-rich sequences under high salt or torsional stress. Question 2. CSIRNet Deck 2016. Which sugar puckering is associated with the A form of DNA and RNA? A. C2 endo. B. C3 endo. C. O4 exo. D. C1 endo. Answer. B. C3 endo. Explanation. The A form helix has a C3 endo sugar pucker, contributing to its compact and tilted structure. Question 3. CSIRNet June 2014. Which of the following RNA structures is involved in tertiary interactions? A. Hairpin loop. B. Internal loop. C. Pseudoknot. D. Stem loop. Answer. C. Pseudoknot. Explanation. Pseudoknots are tertiary structures that contribute to the complex folding and function of RNA. Conclusion. The conformation of nucleic acids is foundational to all biological processes involving genetic material. The ability of DNA and RNA to adopt different forms, shapes, and folds not only supports their functions in a cell but also allows for complex regulation and evolution of gene expression. For CSIRNet Life Sciences, questions on this topic typically probe your understanding of structural details, differences between A, B, and Z forms, and the biological relevance of RNA folding. Mastery of these concepts is essential for tackling biochemistry and molecular biology questions in the exam. Let me know your next topic for notes. I'll keep building your JRF level masterclass step by step. You said, Helix A, ChatGPT said. Helix types in nucleic acids. A. Helix CSIRNet JRF Life Sciences. In-depth theory note paragraph format max word limit no tables previous year questions included. The A helix, also referred to as A4 nucleic acid, is one of the three major helical forms that double-stranded nucleic acids can adopt. The conformation of DNA or RNA into A form is heavily influenced by hydration levels, ion concentration, and the nature of the sugar ribose or deoxyribose. In biological systems, the A form is not the predominant conformation for double-stranded DNA but is commonly found in RNA duplexes and DNA-RNA hybrids. Structurally, the A helix is a right-handed helix, like the B helix, the most common form of DNA, but it is shorter and broader. It comprises approximately 11 base pairs per turn, in contrast to the 10.5 BP per turn of bDNA. The helical pitch distance for complete turn is about 28.2 A, and the diameter of the helix is around 26 A, making it appear more compact and dehydrated in comparison to bDNA. The sugar pucker of the nucleotide in A form helix adopts a C3' endo conformation, which results in a different spatial arrangement of the phosphate backbone and the base pairs. This puckering is favored in RNA due to the presence of the 2' group on the ribose sugar, which sterically hinders the C2' endo pucker typical of B form DNA. Consequently, RNA cannot easily adopt the B form, and thus prefers the A form when double-stranded. One of the most distinct features of the A form helix is the tilting of the base pairs. The bases are tilted approximately 20 degrees away from the perpendicular axis, and they are displaced from the central axis of the helix. This tilt and displacement cause the formation of a deep, narrow major groove and a shallow, wide minor groove, a structural arrangement that significantly influences how proteins and ligands interact with the helix. aDNA, the dehydrated form of DNA, can form under low humidity conditions or in the presence of alcohols or cations that neutralize phosphate charges. In vitro, X-ray crystallography often reveals A form helices, especially in oligonucleotides crystallized under such dehydrating conditions. Importantly, DNA-RNA hybrids formed during transcription and double-stranded RNA, dsRNA, naturally adopt the A form in physiological conditions. The structure is thus central to many biological processes, including RNA interference, Turner recognition, and CRISPR SAW mechanisms. From an evolutionary perspective, the ability of RNA to stably form the A form helix despite being single-stranded underpins its versatility as both genetic material and catalyst in early life forms. This aligns with the RNA world hypothesis, which posits that RNA predated DNA and proteins in early evolution. Previous year CSIRNet questions. Question 1. CSIRNet Deck 2016. What is the sugar puckering found in the A form of nucleic acid helix? A. C2' endo. B. C3' endo. C. C1 endo. D. O4' exo. Answer. B. C3' endo. Explanation. The A form helix is characterized by C3' endo sugar pucker, commonly found in RNA due to the presence of the 2' group. Question 2. CSIRNet June 2018. Which of the following is true regarding A form helix? A. It is a left-handed helix with 10 base pairs per turn. B. It is a right-handed helix with tilted base pair. C. It has a flat major groove and wide minor groove. D. It is the most common DNA conformation in vivo. Answer. B. It is a right-handed helix with tilted base pairs. Explanation. A form helix is right-handed with bases tilted from the helical axis, distinguishing it from the upright base orientation in B form. Question 3. CSIRNet June 2017. RNA molecules adopt A form helices because of the presence of uracil promotes A helix B. RNA cannot form hydrogen bond C. The 2' group in RNA favors C3' endo pucker. D. RNA is always double-stranded. Answer. C. The 2' group in RNA favors C3' endo pucker. Explanation. The steric hindrance of the 2' group in RNA prevents C2' endo pucker and promotes the C3' endo sugar conformation of the A helix. Conclusion. Understanding the A form helix is critical in decoding RNA structure and function. Its geometry, base tilting, and groove architecture influence molecular recognition, stability, and catalytic activity in nucleic acid-based processes. For CSIRNet Life Sciences, precise knowledge of structural differences between A, B, and Z forms, especially in the context of sugar puckering and helical properties, is essential to solve conceptual questions and diagrams. Expect these as direct MCQs or in matching or assertion reason formats in the exam. Let me know if you'd like the next one on B helix, Z helix, or move forward in your CSIR syllabus. You said, B. ChatGPT said. Helix types in nucleic acids. B helix B form DNA. CSIRNet JRF Life Sciences. Full length theory note paragraph format. Maximum word limit. No tables. Previous year questions included. The B helix or B form DNA is the most stable and prevalent form of double-stranded DNA under physiological conditions. First elucidated by Watson and Crick in 1953 using Rosalind Franklin's X-ray diffraction data. This form is the canonical right-handed helix structure of DNA observed in cells. It plays a central role in genomic packaging, replication, transcription, and protein DNA interactions, making its understanding foundational for molecular biology and CSIRNet Life Sciences preparation. The B helix has a right-handed helical structure with approximately 10.5 base pairs per turn and a helical pitch of 34 A. The diameter of the helix is about 20 A. Each base pair is nearly perpendicular to the helical axis, and the base stacking provides hydrophobic interactions which are essential for the stability of the helix. Hydrogen bonding between complementary base pairs AT with 2H bonds, GC with 3H bonds stabilizes the structure, but base stacking is a major contributor to thermodynamic stability. The sugar in bDNA adopts the C2' endo pucker, distinguishing it from A form nucleic acids, which have a C3' endo conformation. This puckering affects the orientation of the phosphate backbone and hence the overall geometry of the double helix. The major groove in bDNA is wide and deep, allowing for sequence-specific protein recognition, while the minor groove is narrower and shallower. Another defining feature is the anti-conformation of the nitrogenous bases with respect to the sugar, which enables optimal base pairing and stacking. The backbone conformation follows a zigzag path as it alternates between sugars and phosphate groups. The helix is stabilized by electrostatic interactions involving the negatively charged phosphate groups and associated cations like Mg² or polyamines. bDNA exists predominantly in hydrated environments, making it the native form of DNA in most living cells. The structural integrity of B-form DNA is affected by ionic strength, humidity. The B helix, or B-form DNA, is the most stable and prevalent form of double-stranded DNA under physiological conditions. First elucidated by Watson and Crick in 1953 using Rosalind Franklin's X-ray diffraction data, this form is the canonical right-handed helix structure of DNA observed in cells. It plays a central role in genomic packaging, replication, transcription, and protein DNA interactions, making its understanding foundational for molecular biology and CSIR-net life sciences preparation. The B helix has a right-handed helical structure with approximately 10.5 base pairs per turn and a helical pitch of the B helix, or B-form DNA, is the most stable and prevalent form of double-stranded DNA under physiological conditions. First elucidated by Watson and Crick in 1953 using Rosalind Franklin's X-ray diffraction data, this form is the canonical right-handed helix structure of DNA observed in cells. The B helix, or B-form DNA, is the most stable and prevalent form of double-stranded DNA under physiological conditions. First elucidated by Watson and Crick in 1953 using Rosalind Franklin's X-ray diffraction data, this form is the canonical right-handed helix structure of DNA observed in cells. It plays a central role in genomic packaging, replication, transcription, and protein DNA interactions, making its understanding foundational for molecular biology and CSIR-net life sciences preparation. The B helix has a right-handed helical structure with approximately 10.5 base pairs per turn and a helical pitch of 34A. The diameter of the helix is about 20A. Each base pair is nearly perpendicular to the helical axis, and the base stacking provides hydrophobic interactions which are essential for the stability of the helix. Hydrogen bonding between complementary base pairs AT with 2H bonds, GC with 3H bonds stabilizes the structure, but base stacking is a major contributor to thermodynamic stability. The sugar in bDNA adopts the C2' endo pucker, distinguishing it from E4 nucleic acids, which have AC3' endo conformation. This puckering affects the orientation of the phosphate backbone and hence the overall geometry of the double helix. The major groove in bDNA is wide and deep, allowing for sequence-specific protein recognition, while the minor groove is narrower and shallower. Another defining feature is the anti-conformation of the nitrogenous bases with respect to the sugar, which enables optimal base pairing and stacking. The backbone conformation follows a zigzag path as it alternates between sugars and phosphate groups. The helix is stabilized by electrostatic interactions involving the negatively charged phosphate groups and associated cations like Mg² or polyamines. bDNA exists predominantly in hydrated environments, making it the native form of DNA in most living cells. The structural integrity of B-form DNA is affected by ionic strength, humidity, sequence composition, and intercalating agents. Sequences rich in G as defined as C pairs exhibit greater thermal stability due to stronger hydrogen bonding and tighter base stacking. The B form is flexible, allowing for local deviations from ideal geometry, which is critical during DNA replication, repair, and transcription. DNA binding proteins, such as transcription factors, polymerases, and histones, interact with specific base sequences via the major groove, relying on the predictable geometry of the B helix. Nucleosome formation also depends on the bending flexibility of bDNA, wrapping around histone cores in chromatin. From a functional perspective, the stability and regularity of the B form are essential for faithful replication, efficient repair mechanisms, and accurate transcription. Variations from the B form, like transitions to A form or Z form, often occur transiently during biological processes and can be involved in regulatory mechanisms. Previous year CSIR-net questions. Question 1. CSIR-net Deck 2017. Which of the following features is characteristic of B form DNA? A. Left-handed helix with 12 BP per turn. B. Right-handed helix with C2' endo sugar pucker. C. Left-handed helix with C3' endo sugar pucker. D. Right-handed helix with 11 BP per turn and deep minor groove. Answer. B. Right-handed helix with C2' endo sugar pucker. Explanation. The B form is right-handed and features AC2' endo sugar conformation, common in DNA under physiological conditions. Question 2. CSIR-net June 2020. In B form DNA, which of the following is true about the base pairing and helical parameters? A. Bases are tilted and displaced from the axis. B. Helix has 10.5 BP. Turn and base pairs are nearly perpendicular to the axis. C. Helical diameter is in GT 26A. D. DNA exists in B form under dehydrated conditions. Answer. B. Helix has 10.5 BP. Turn and base pairs are nearly perpendicular to the axis. Explanation. This is a hallmark of bDNA geometry. 10.5 base pairs per turn with base pairs almost perpendicular to the helical axis. Question 3. CSIR-net Deck 2016. Which of the following grooves in bDNA is more accessible for transcription factors? A. Minor groove. B. Major groove. C. Both equally. D. Grooves are not important for protein interaction. Answer. B. Major groove. Explanation. The major groove is wider and provides a more accessible surface for transcription factors and other regulatory proteins. Conclusion. The B form DNA is central to molecular biology and genetics. For CSIR-net life sciences, questions on bDNA typically test your understanding of helical parameters, chemical and geometric conformations, base pairing rules, and functional implications. A precise grasp of how bDNA interacts with proteins and changes its structure under different biological conditions is essential. These aspects are repeatedly tested and should be mastered thoroughly. Always associate bDNA with standard genomic DNA structure, major groove recognition, C2' endo sugar puckering, and right-handed helicity in your revision. Let me know when you're ready for zDNA, or if you'd like the next topic in the syllabus. You said Z. ChatGPT said Z helix Z form DNA. CSIR-net JRF Life Sciences full-length theory lecture paragraph format, no tables, maximum detail, previous year questions with answers in here. The Z helix, or Z form DNA, is a left-handed double helical structure of DNA that represents an alternative conformation to the more common B form. It is named Z because of the zigzag backbone path of the phosphodiester linkage when viewed from the side. zDNA is a rare but biologically significant conformation that typically forms in sequences rich in alternating purine pyrimidine repeats, especially GCN or GTN sequences, under high salt or supercoiled conditions. It has been implicated in the regulation of gene expression, DNA recombination, and chromatin remodeling. In contrast to bDNA, which is a right-handed helix, zDNA has a left-handed twist, which results in a dramatically different geometry. The Z helix consists of 12 base pairs per turn and has a pitch of approximately 45A, making it longer and thinner than the B form. Its helical diameter is about 18A. Unlike the B form, in which all bases adopt the anti-conformation, in zDNA, the purines typically guanine adopt the syn conformation, while the pyrimidines cytosine remain in the anti-conformation. This alternation leads to the zigzag phosphate backbone appearance. The sugar puckering in zDNA also alternates. Guanine residues adopt AC3' endo sugar conformation, and cytosine residues adopt AC2' endo pucker. This switch in sugar pucker and base conformation introduces strain and makes zDNA less stable than bDNA under normal physiological conditions. However, the Z form can become stabilized in high ionic strength, methylation of cytosine residues, or negative supercoiling, especially during transcription where local unwinding occurs. zDNA has biological relevance in eukaryotic genomes. It is transiently formed behind active RNA polymerase during transcription due to negative supercoiling. The formation of zDNA in these regions may act as a torsional stress relief mechanism and might be involved in modulating gene expression by providing binding sites for zDNA binding proteins like the DAR1 adenosine deaminase acting on RNA1. These proteins can recognize the unique zigzag shape of zDNA, enabling regulatory interactions. zDNA is also implicated in epigenetic regulation and has been found in regions rich in CpG dinucleotides, especially when the cytosine is methylated. Methylation enhances the propensity for zDNA formation, linking it to chromatin structure and potential gene silencing or activation events. Moreover, the presence of zDNA can act as a marker of genomic instability, and its formation is associated with certain neurological and autoimmune diseases. Structurally, zDNA does not have well-defined major or minor grooves like bDNA. Instead, it features a deep and narrow minor groove and a barely noticeable major groove, which further differentiates it in terms of protein-DNA interaction and accessibility. This unique groove profile contributes to its selectivity in binding specific proteins and enzymes that recognize zDNA exclusively. Previous year CSIR-net questions. Question 1. CSIR-net June 2018. Which of the following features distinguishes zDNA from bDNA? A. Right-handed helix with anti-conformation of bases B. Left-handed helix with alternating sin and anti-conformation C. Right-handed helix with C3' endo-sugar pucker D. Uniform anti-conformation of all nucleotides. Answer. B. Left-handed helix with alternating sin and anti-conformations. Explanation. zDNA is a left-handed helix characterized by sin conformation of guanine and anti-conformation of cytosine. Question 2. CSIR net deck 2019. zDNA is more likely to form in regions that are A. AT rich under high humidity B. GC rich with methylated cytosines under supercoiling C. Random sequences in relaxed DNA D. Polyattracts in nucleosomes. Answer. B. GC rich with methylated cytosines under supercoiling. Explanation. zDNA forms preferentially in GC-rich regions, especially when cytosines are methylated and DNA is negatively supercoiled. Question 3. CSIR net June 2021. zDNA binding proteins preferentially interact with a major groove of bDNA. B. Minor groove of aDNA C. Zigzag phosphate backbone of zDNA D. Unwound regions of DNA replication forks. Answer. C. Zigzag phosphate backbone of zDNA. Explanation. Proteins like the DAR1 bind the unique zigzag backbone of zDNA, not the grooves like in B form. Conclusion. zDNA may be a minor structural form, but its role in gene regulation, chromatin structure, epigenetics, and protein interaction makes it a high-priority topic in the CSIR net syllabus. Understanding its distinct left-handed conformation, zigzag backbone, sin anti-base orientations, and conditions favoring its formation is essential. As CSIR net questions often focus on comparing structural and functional aspects of DNA forms, particularly Z versus B form, being fluent with these differences can offer a scoring advantage in the exam. Let me know when you're ready for the next topic, aDNA or anything else from your CSIR net life sciences syllabus. You said, tRNA, ChatGPT said, tRNA, transfer RNA. CSIR net JRF life sciences, full-length theory lecture, paragraph format, no tables, maximum detail, previous year questions with answers in year. Transfer RNA, tRNA, is a crucial molecular component involved in the process of translation, facilitating the conversion of genetic information into protein. Each TRNA molecule serves as an adapter, linking the mRNA codon to the corresponding amino acid through anticodon recognition. This process is fundamental to protein synthesis, making TRNA an essential player in genetic expression. For CSIR net life sciences, understanding the structure, function, and role of TRNA in translation is crucial for mastering molecular genetics. A typical TRNA molecule has a cloverleaf structure with 4 distinct arms. The amino acid arm or acceptor stem, which is the site for amino acid attachment. The anticodon arm, which contains the anticodon sequence that recognizes the complementary mRNA codon. The D arm, containing the dihydroiodine D loop, involved in structural stability. The T arm, with the T-side C loop containing ribithymidine, pseudouridine, and cytosine important for interactions with ribosomal RNA, rRNA during translation. In its tertiary structure, TRNA adopts an L shape, formed by the anticodon arm and amino acid arm, which are positioned to interact with the ribosome during protein synthesis. The structure is stabilized by base pairing interactions within the stem loops, and hydrogen bonds between the backbone and base pairs ensure its stability and functionality. The 3' end of the TRNA molecule is where the amino acid is covalently attached via an ester bond. This attachment is catalyzed by amino acyl TRNA synthetases, enzymes that are highly specific for each amino acid and its corresponding TRNA. The genetic code is degenerate, meaning that more than one codon can code for the same amino acid. Each TRNA molecule can recognize multiple codons through wobble pairing at the third end of the codon. This flexibility in codon recognition is provided by the wobble hypothesis, which allows some TRNA molecules to recognize more than one codon for the same amino acid. The primary function of TRNA is to deliver amino acids to the ribosome during translation. The ribosome reads the mRNA codons, and TRNA molecules bring the corresponding amino acids, which are then added to the grown polypeptide chain. The anticodon of the TRNA binds to the complementary codon on the mRNA, ensuring that the correct amino acid is incorporated into the polypeptide according to the genetic code. In addition to its role in translation, TRNA also plays a part in proofreading during protein synthesis. The specificity of amino acylation, the attachment of the correct amino acid to TRNA, is key to maintaining the fidelity of translation. If the wrong amino acid is attached to a TRNA, the ribosome will incorporate an incorrect amino acid into the grown protein chain, which can lead to malfunctioning proteins. Hence, amino acyl TRNA synthetases have proofreading mechanisms to ensure that the correct amino acid is charged to the TRNA. Previous year CSIR net questions. Question 1. CSIR net June 2017. Which of the following is the correct sequence of events in the process of TRNA charging? A. Amino acid binds to TRNA, right pointing arrow TRNA binds to ribosome, right pointing arrow protein synthesis occurs. B. Amino acid is activated by amino acyl TRNA synthetase, right pointing arrow TRNA binds amino acid, right pointing arrow TRNA binds ribosome. C. TRNA binds to mRNA, right pointing arrow amino acid binds to TRNA, right pointing arrow ribosome catalyzes protein synthesis. D. Ribosome catalyzes. Answer. B. Amino acid is activated by amino acyl TRNA synthetase, right pointing arrow TRNA binds amino acid, right pointing arrow TRNA binds ribosome. Explanation. The correct sequence involves the activation of the amino acid by amino acyl TRNA synthetase, followed by the binding of the amino acid to TRNA, and finally the TRNA binding to the ribosome during translation. Question 2. CSIR net deck 2018. Which part of TRNA directly interacts with the mRNA codon during translation? A. T arm. B. D arm. C. Anticodon loop. D. Amino acid arm. Answer. C. Anticodon loop. Explanation. The anticodon loop of the TRNA contains the anticodon sequence, which binds to the complementary codon on the mRNA during translation. Question 3. CSIR net June 2019. The wobble hypothesis explains. A. The flexibility. Answer. Of the flexibility in codon recognition by TRNA at the third position of the codon. Explanation. The wobble hypothesis explains that the third position of the mRNA codon can be recognized by a single TRNA, allowing multiple codons to specify the same amino acid. Conclusion. TRNA is an essential molecule in the translation process, where it bridges the genetic code carried by mRNA with the correct amino acid sequence for protein synthesis. Understanding its structure, amino acid charging, anticodon codon recognition, and role in maintaining translation fidelity is key for mastering molecular genetics concepts in CSIR net life sciences. Focus on the functional aspects of TRNA, its interactions with amino acyl TRNA synthetase, and its contributions to protein synthesis efficiency. Let me know when you're ready for the next topic. You said, microRNA. ChatGPT said, microRNA. Myrna. CSIR net JRF life sciences, full-length theory lecture, paragraph format, no tables, maximum detail, previous year questions with answers in year. MicroRNAs are a class of small, non-coding RNAs that play a pivotal role in regulating gene expression at the post-transcriptional level. These molecules, typically around 22 nucleotides in length, are involved in fine-tuning the expression of a broad range of genes by binding to complementary sequences in the 3' untranslated regions, 3 UTRs of target mRNAs. mRNAs can either induce mRNA degradation or inhibit translation, depending on the degree of complementarity between the mRNA and its target mRNA. mRNAs are initially transcribed as part of a larger primary mRNA transcript, pre-mRNA, by RNA polymerase II. This long transcript undergoes processing in the nucleus by the Drosha DGCR8 complex, which cleaves the pre-mRNA to produce a precursor mRNA, pre-mRNA. The pre-mRNA is then exported from the nucleus to the cytoplasm by the EXPORTIN5 protein. Once in the cytoplasm, the pre-mRNA is processed further by the dicer enzyme, which cleaves it into a duplex of mRNA and its passenger strand. The guide strand of the duplex is incorporated into the RNA-induced silencing complex, RISC, while the passenger strand is typically degraded. The RISC complex then mediates the regulatory function of the mRNA by guiding the guide strand to the target mRNA. If there is perfect or near perfect complementarity between the mRNA and the mRNA is typically cleaved and degraded. If the complementarity is partial, as is more often the case, the mRNA causes translation repression rather than mRNA degradation. This translation inhibition occurs through mechanisms such as preventing ribosome assembly on the mRNA or causing dead-end mRNA, which is stalled in translation initiation. mRNAs are involved in regulating a wide variety of biological processes, including development, cell differentiation, proliferation, apoptosis, and stress responses. In particular, they are crucial for the fine-tuning of gene expression in various pathways, including those controlling cell cycle progression, immune responses, metabolism, and neurogenesis. mRNAs also have significant roles in cancer, where the dysregulation can lead to either oncomeres, mRNAs that promote cancer, or tumor suppressor mRNAs. The biogenesis and regulation of mRNAs are highly controlled, with the expression of specific mRNAs being modulated by transcription factors, epigenetic modifications, and RNA binding proteins. Furthermore, the mRNA interaction is not always one-to-one. A single mRNA can regulate multiple target mRNAs, and a single mRNA may be regulated by multiple mRNAs, contributing to a complex regulatory network. The discovery of mRNAs was groundbreaking because it showed that non-coding RNAs, long thought to be irrelevant, could have profound regulatory functions in the cell. This has opened up a new avenue for therapeutic strategies, including the use of mRNA mimics or inhibitors to modulate gene expression in diseases like cancer, neurodegeneration, and viral infections. Previous year CSIRNet questions. Question 1. CSIRNet Deck 2017. Which of the following is the correct mechanism of action of mRNAs? A. mRNAs directly bind to the ribosome and enhance protein synthesis. B. mRNAs bind to mRNA and promote its translation. C. mRNAs bind to mRNA and induce its degradation or inhibit translation. D. mRNAs activate the transcription of target genes. Answer. C. mRNAs bind to mRNA and induce its degradation or inhibit translation. Explanation. mRNAs typically induce mRNA degradation or inhibit translation by binding to complementary sequences in the 3' UTR of the target mRNA. Question 2. CSIRNet June 2019. The primary function of the RISC complex in the context of mRNA regulation is to process. Answer. C. To guide the mRNA to its target mRNA for regulation. Explanation. The RISC complex is responsible for guiding the mRNA to its target mRNA, where it can induce degradation or inhibit translation. Question 3. CSIRNet Deck 2020. Which of the following best describes the biogenesis of mRNAs? A. mRNAs are transcribed by RNA polymerase I and processed by RNase 3. B. mRNAs are transcribed as a precursor that is processed. Answer. B. mRNAs are transcribed as a precursor that is processed into a mature mRNA by dicer. Explanation. mRNAs are initially transcribed as primary mRNAs, pre-mRNAs, then processed into precursor mRNAs, pre-mRNAs by Drosha, and finally processed into mature mRNAs by dicer in the cytoplasm. Conclusion. mRNAs are essential for the fine regulation of gene expression in cells, controlling processes such as development, apoptosis, and stress responses. Their biogenesis, involving RNA polymerase II, Drosha, dicer, and RISC, plays a crucial role in their function. Understanding the mechanisms by which mRNAs influence mRNA stability and translation is essential for mastering cellular regulatory pathways and preparing for the CSIRNet Life Sciences exam. The implications of mRNA dysregulation in diseases like cancer and neurodegeneration further underscore their importance in biological research and therapeutic strategies. Let me know when you're ready for the next topic. You said. Stability of proteins. ChatGPT said. Stability of proteins. CSIRNet JRF Life Sciences. Protein stability refers to the ability of a protein to maintain its functional conformation under physiological conditions. This concept is crucial in understanding protein folding, denaturation, and the roles proteins play within living organisms. Stability is essential for proper cellular functions, and the failure of proteins to fold correctly can lead to diseases like cystic fibrosis, Alzheimer's, and prion diseases. In the context of CSIRNet Life Sciences, it is critical to comprehend the factors that contribute to protein stability and the mechanisms that govern their proper folding and degradation. The native conformation of a protein is its most thermodynamically stable state, the configuration with the lowest free energy. Proteins achieve their stable conformations through a series of non-covalent interactions, including hydrophobic interactions, hydrogen bonding, electrostatic interactions, and van der Waals forces. These interactions collectively determine the three-dimensional structure that allows proteins to function in specific biological processes. The stability of proteins is influenced by environmental factors such as temperature, pH, ionic strength, and the presence of denaturing agents like urea or guanidine hydrochloride. Hydrophobic interactions are the most significant contributors to protein stability. In an aqueous environment, hydrophobic residues tend to cluster together in the interior of the protein, away from the solvent, whereas hydrophilic residues are exposed on the exterior, interacting with water. This creates a hydrophobic core, which is essential for the stability of the protein's folded structure. Hydrogen bonds further stabilize the secondary structure, such as alpha helices and beta sheets, by protein stability refers to the ability of a protein to maintain its functional conformation under physiological conditions. This concept is crucial in understanding protein folding, denaturation, and the roles proteins play within living organisms. Stability is essential for proper cellular functions, and the failure of proteins to fold correctly can lead to diseases like cystic fibrosis, Alzheimer's, and prion diseases. In the context of CSIRNet Life Sciences, it is critical to comprehend the factors that contribute to protein stability and the mechanisms that govern their proper folding and degradation. The native conformation of a protein is its most thermodynamically stable state, the configuration with the lowest free energy. Proteins achieve their stable conformations through a series of non-covalent interactions, including hydrophobic interactions, hydrogen bonding, electrostatic interactions, and van der Waals forces. These interactions collectively determine the three-dimensional structure that allows proteins to function in specific biological processes. The stability of proteins is influenced by environmental factors such as temperature, pH, ionic strength, and the presence of denaturing agents like urea or guanidine hydrochloride. Hydrophobic interactions are the most significant contributors to protein stability. In an aqueous environment, hydrophobic residues tend to cluster together in the interior of the protein, away from the solvent, whereas hydrophilic residues are exposed on the exterior, interacting with water. This creates a hydrophobic core, which is essential for the stability of the protein's folded structure. Hydrogen bonds further stabilize the secondary structure, such as alpha helices and beta sheets, by maintaining the regularity of the backbone atoms. Electrostatic interactions and salt bridges between oppositely charged residues also contribute to the stability, particularly in the protein's tertiary structure. However, protein stability is not static, it is a dynamic equilibrium between the folded and unfolded states. Under unfavorable conditions, proteins may undergo denaturation, a process where they lose their native conformation and, as a result, their biological activity. Denaturation can be caused by several factors, including high temperatures, extreme pH, or chemical denaturants. Denatured proteins often expose hydrophobic regions, which can lead to aggregation, a characteristic feature of many neurodegenerative diseases. To counteract protein misfolding, cells employ chaperones and chaperonins, specialized proteins that assist in the correct folding of other proteins. Molecular chaperones, such as heat shock proteins, HSPs, facilitate the refolding of denatured proteins and prevent aggregation by binding to exposed hydrophobic regions. Additionally, the proteasome system and autophagy pathways are involved in degrading damaged or misfolded proteins, ensuring cellular homeostasis. Proteins must also exhibit kinetic stability, meaning they must not unfold or aggregate prematurely during cellular processes such as translation and transport. A protein stability is linked to its half-life, the time it takes for half of the protein molecules to be degraded. This process is regulated by specific signal peptides or tags such as the ubiquitin proteasome system for marking proteins for degradation. In summary, protein stability is determined by a delicate balance of intrinsic forces within the protein itself and extrinsic factors such as environmental conditions. Misfolding, denaturation, and aggregation of proteins are associated with numerous diseases, highlighting the importance of protein stability in maintaining cellular functions. The regulatory systems in cells, including chaperones and proteolytic systems, ensure that proteins remain functional and avoid malfunction. Previous year CSIRNet questions. Question 1. CSIRNet June 2018. Which of the following is the primary factor responsible for protein denaturation? A. Decrease in temperature. B. Changes in pH. C. Presence of molecular chaperones. D. High concentration of salts. Answer. B. Changes in pH. Explanation. Changes in pH can disrupt the hydrogen bonds and electrostatic interactions that stabilize the protein's tertiary and quaternary structures, leading to denaturation. Question 2. CSIRNet June 2019. Which of the following is not a factor influencing the stability of a protein? A. Ionic strength. B. Hydrophobic interaction. C. Ribosome synthesis rate. D. Hydrogen bonding. Answer. C. Ribosome synthesis rate. Explanation. The ribosome synthesis rate does not directly affect protein stability. However, ionic strength, hydrophobic interactions, and hydrogen bonding are crucial factors that contribute to protein stability. Question 3. CSIRNet Deck 2020. The half-life of a protein in a cell primarily depends on the rate of protein synthesis. B. The protein's amino acid sequence and degradation signal. C. The concentration of ions in a cell. D. The temperature of the environment. Answer. B. The protein's amino acid sequence and degradation signals. Explanation. The half-life of a protein is regulated by its amino acid sequence, which can contain degradation signals such as the ubiquitin tag, marking it for degradation by the proteasome. Conclusion. Protein stability is essential for maintaining cellular function and integrity. The proper folding of proteins, driven by various non-covalent interactions, ensures their functional conformation. Protein misfolding, denaturation, and aggregation can have severe consequences, such as the onset of diseases like Alzheimer's and Parkinson's. The molecular chaperones, and proteolytic systems help maintain protein stability by assisting in folding and removing damaged proteins. Understanding the factors that influence protein stability is critical for CSIRNet Life Sciences and is essential for developing therapeutic approaches to treat protein-related diseases. Let me know when you're ready for the next topic. You said. Stability of nucleic acids. ChatGPT said. Stability of nucleic acids. CSIRNet JRF Life Sciences full-length theory lecture, paragraph format, no tables, maximum detail, previous year questions with answers in here. Nucleic acid stability refers to the ability of DNA and RNA to maintain their functional structures and biological activities under physiological conditions. The stability of nucleic acids is critical for proper genetic information storage, replication, transcription, and translation. Both DNA and RNA molecules must withstand various factors that could otherwise cause denaturation, degradation, or mutations that affect their function and integrity. The primary structure of nucleic acids, the sequence of nucleotides, is inherently stable due to the phosphodiester bonds between the sugar and phosphate groups of adjacent nucleotides. However, the secondary and tertiary structures of nucleic acids, such as the double helix of DNA and the hairpin loops of RNA, are more susceptible to changes in their environment. Therefore, DNA and RNA stability is influenced by factors such as temperature, ionic strength, pH, and the presence of nucleases enzymes that degrade nucleic acids. DNA stability. The stability of DNA primarily comes from the hydrogen bonding between the purine and pyrimidine bases, specifically the AT and GC base pairs. The double-stranded structure of DNA creates a stable molecule because the hydrophobic bases are buried in the interior, away from the aqueous environment, while the hydrophilic sugar phosphate backbone is exposed to the solvent. The GC base pairs, with 3 hydrogen bonds compared to the AT pairs 2, confer greater stability to the DNA molecule. The stability of DNA is also influenced by its supercoiling and topological state, which can be regulated by topoisomerases. These enzymes prevent DNA breakage during processes like replication and transcription by introducing controlled supercoiling. In addition, protective proteins such as histones in eukaryotes or HU proteins in bacteria can stabilize DNA by binding to the molecule and helping it to maintain its structure under stress conditions. Environmental conditions also affect DNA stability. High temperatures can lead to denaturation, where the double-stranded DNA separates into single strands, disrupting hydrogen bonds. Extreme pH levels can alter the charge on the phosphate backbone, weakening the structure. Additionally, UV radiation and chemical mutagens can cause base pair mismatches, thymine dimers, and other forms of DNA damage that compromise stability. RNA stability. RNA is typically less stable than DNA due to its single-stranded nature and the presence of the hydroxyl group at the 2' position of the ribose sugar, which makes the phosphodiester bonds more prone to hydrolysis. Additionally, the secondary structures of RNA, such as hairpin loops and pseudoknots, are highly dynamic, which can lead to denaturation or misfolding if not properly stabilized. RNA molecules are typically more labile than DNA and are subjected to rapid degradation in the cell. The stability of RNA is regulated by its 5' cap in eukaryotes, which protects the 5' end from exonucleolytic degradation, and the polyA tail at the 3' end, which influences the half-life of the RNA molecule. In addition, RNA-binding proteins, such as RNA helicases and exonucleases, play important roles in regulating RNA stability by either promoting or inhibiting degradation. In prokaryotes, RNA stability is often influenced by the 5' to 3' exonuclease activity of enzymes such as RNase, which degrades RNA from the 5' end. In contrast to the relatively short-lived RNA, our RNA internal molecules are highly stable due to their secondary and tertiary structures, which provide protection from degradation. For instance, ternars are often stabilized by extensive base pairing within their secondary structure, making them more resistant to degradation by exonucleases. Factors affecting nucleic acid stability. Temperature. High temperatures cause the denaturation of nucleic acids by breaking the hydrogen bonds between complementary bases in the helix. DNA melting temperature (TM) is a temperature at which half of the DNA molecules become denatured, and this depends on the GC content, with GC-rich DNA having a higher TM. Ionic strength. The presence of salt helps stabilize the DNA structure by neutralizing the negative charges on the phosphate backbone, which otherwise would repel each other. However, low ionic strength leads to destabilization and denaturation of nucleic acids. pH. Extreme pH levels can alter the protonation state of nucleotides, disrupting base pairing and overall structure. Alkaline conditions can lead to hydrolysis of the phosphodiester bond in RNA, making it less stable. Enzymatic degradation. The presence of nucleases, such as DNAs and RNAs, can degrade nucleic acids, making their stability dependent on the presence of protective factors like protein binding and capping. UV radiation. UV light induces pyrimidine dimers, thymine dimers in DNA, causing mutations that destabilize the DNA helix. Protection mechanisms. To maintain nucleic acid stability, cells have several protective mechanisms. For DNA, there are repair mechanisms like base excision repair (BER), nucleotide excision repair (NER), and mismatch repair (MMR) that correct DNA damage and maintain the integrity of the genetic material. Additionally, the presence of protective proteins, such as histones in eukaryotes, helps shield DNA from external damage. For RNA, there is mRNA surveillance where nonsense-mediated decay (NMD) or mRNA decay pathways recognize and degrade faulty or incorrectly processed RNA molecules, preventing the expression of dysfunctional proteins. Previous year CSIRNet questions. Question 1. CSIRNet June 2017. Which of the following factors does not affect the stability of DNA? A. Ionic strength. B. UV radiation. C. Base composition. D. Enzyme concentration. Answer. D. Enzyme concentration. Explanation. While ionic strength, UV radiation, and base composition influence DNA stability, enzyme concentration does not directly affect the stability of DNA unless it involves nuclease activity. Question 2. CSIRNet Deck 2018. What is the role of the 5' cap structure? What is the role of the 5' cap structure in eukaryotic mRNA stability? A. It protects the mRNA from exonucleolytic. Answer. It protects the mRNA from exonucleolytic degradation. Explanation. The 5' cap structure prevents exonucleolytic degradation of mRNA by protecting the 5' end from being attacked by exonucleases. Question 3. CSIRNet June 2019. The melting temperature (TM) of a DNA molecule is affected by a. The size of the molecule only. B. The concentration of salts. C. The secondary structure of the RNA. D. The enzyme activity during transcription. Answer. B. The concentration of salts. Explanation. Salt concentration stabilizes the DNA helix by neutralizing the repulsive forces between the negatively charged phosphate groups. The melting temperature (TM) is higher for DNA with a higher GC content and in high salt concentrations. Conclusion. The stability of nucleic acids is fundamental to cellular function and genetic integrity. Factors such as temperature, pH, ionic strength, and the presence of protective proteins and repair mechanisms ensure that both DNA and RNA maintain their structure and function. Disruptions to nucleic acid stability can lead to mutations, genetic diseases, and cellular dysfunctions. Understanding the factors that influence nucleic acid stability is essential for mastering the concepts required for the CSIRNet Life Sciences exam. Let me know when you're ready for the next topic. You said. Metabolism of carbohydrates. ChatGPT said. Metabolism of carbohydrates. CSIRNet JRF Life Sciences full-length theory lecture, paragraph format, no tables, maximum detail, previous year questions with answers in here. Carbohydrates are one of the most important classes of biological macromolecules, serving as a primary source of energy and playing critical roles in cellular structure and signaling. The metabolism of carbohydrates encompasses the biochemical pathways that process carbohydrates for energy production, storage, and synthesis of other biomolecules. It includes both catabolic and anabolic pathways that regulate the breakdown of sugars, such as glucose, and the synthesis of complex carbohydrates, such as glycogen and starch. 1. Glycolysis. Glycolysis is a catabolic pathway that breaks down glucose, a 6-carbon sugar, into pyruvate, a 3-carbon compound with the production of ATP and NADH. This pathway occurs in the cytoplasm of the cell and consists of 10 enzyme-catalyzed steps. Glycolysis can proceed under both aerobic and anaerobic conditions. In the energy investment phase, 2 ATP molecules are consumed to phosphorylate glucose and its derivatives, preparing them for later breakdown. In the energy payoff phase, 4 ATP molecules are produced, along with 2 NADH molecules, by substrate-level phosphorylation and the oxidation of glyceraldehyde 3-phosphate. At the end of glycolysis, pyruvate is formed, which may undergo further metabolism depending on the availability of oxygen. Important enzymes: hexokinase, PHOS-PHOFRUCTOKINASE1, PFK1, and pyruvate kinase. Regulation: the key regulatory steps in glycolysis are catalyzed by PFK1 and pyruvate kinase, which are influenced by factors such as ATP, AMP, and fructose 2,6-bisphosphate. These enzymes act as metabolic sensors that ensure the efficient production of ATP when energy demand is high. 2. Pyruvate decarboxylation and citric acid cycle, Krebs cycle. After glycolysis, pyruvate enters the mitochondria, where it undergoes decarboxylation to form acetyl-CoA in a reaction catalyzed by the pyruvate dehydrogenase complex (PDC). This reaction releases one molecule of CO2 and produces NADH. The acetyl-CoA then enters the citric acid cycle, CAC, also known as the Krebs cycle or TCA cycle, where it is oxidized to produce ATP, NADH, and FADH2. Each turn of the cycle involves the condensation of acetyl-CoA with oxaloacetate to form citrate, followed by a series of reactions that regenerate oxaloacetate while producing energy carriers. Key steps: citrate formation. Acetyl-CoA combines with oxaloacetate. Decarboxylation produces CO2 and reduces NAD+ to NADH. Production of FADH2 in a step involving succinate dehydrogenase. For each acetyl-CoA, the cycle produces 3 NADH, 1 FADH2, 1 ATP via GTP, and 2 CO2 molecules. Regulation: the cycle is regulated by the availability of acetyl-CoA, NAD+, and ADP. High levels of ATP inhibit the cycle, while ADP and Ca2+ act as activators. 3. Oxidative phosphorylation and ATP production. The NADH and FADH2 produced during glycolysis, pyruvate decarboxylation, and the citric acid cycle are utilized in oxidative phosphorylation, which occurs in the inner mitochondrial membrane. This process consists of 2 main stages: electron transport chain, etc., and chemiosmosis. Electron transport chain, etc. The NOD and FADH2 donate electrons to the electron transport chain, which is composed of several protein complexes, IIV, and ATP synthase. As electrons move through the chain, protons H+ are pumped across the mitochondrial membrane, creating a proton gradient. Chemiosmosis: the proton gradient created by the etc. drives the synthesis of ATP via ATP synthase as protons flow back into the mitochondrial matrix. The final electron acceptor in the chain is oxygen, which combines with electrons and protons to form water. The total ATP yield from one molecule of glucose can reach up to 38 ATPs under aerobic conditions, assuming NADH is transported into mitochondria efficiently. In anaerobic conditions, the process shifts towards lactic acid fermentation in animals or ethanol fermentation in yeast, which regenerates NAD+ but does not produce additional ATP. 4. Glycogen metabolism. Glycogen, the storage form of glucose, is primarily found in the liver and muscle cells. Glycogen metabolism involves 2 major processes. Glycogen synthesis, glycogenesis, the process by which glucose is added to the glycogen molecule. Glucose 6-phosphate is first converted to glucose 1-phosphate, and then UDP glucose is used to elongate the glycogen chain by adding glucose units. Glycogen breakdown, glycogenolysis. In response to low blood glucose levels or increased energy demand, glycogen is broken down into glucose 6-phosphate by the action of glycogen phosphorylase, which is further converted to glucose for energy production. Regulation: the enzymes involved in both synthesis and breakdown are highly regulated by hormones such as insulin, glucagon, and epinephrine. Insulin promotes glycogenesis, while glucagon and epinephrine promote glycogenolysis. 5. Fermentation. In the absence of oxygen, cells can generate ATP via fermentation pathways. In muscle cells, lactic acid fermentation occurs, converting pyruvate into lactate and regenerating NAD+. In yeast cells, alcoholic fermentation occurs, converting pyruvate into ethanol and CO2, also regenerating NAD+. While fermentation is much less efficient in ATP production compared to oxidative phosphorylation, it allows cells to continue ATP production in anaerobic conditions. Previous year CSIR net questions. Question 1. CSIR net June 2017. Which of the following enzymes is involved in the conversion of glucose to glucose 6-phosphate in the glycolytic pathway? A. Hexokinase. B. Explanation: hexokinase catalyzes the phosphorylation of glucose to glucose 6-phosphate, the first step of glycolysis. Question 2. CSIR net Dec 2018. During glycolysis, the enzyme phosphofructokinase 1 (PFK1) is allosterically inhibited by A. ATP. B. NADHC ADPD AMP. Answer: ATP. Explanation: PFK1 is a key regulatory enzyme in glycolysis. It is allosterically inhibited by high levels of ATP to prevent excessive breakdown of glucose when energy is abundant. Question 3. CSIR net June 2019. Which of the following is the final electron acceptor in the electron transport chain during oxidative phosphorylation? A. NOD. B. Oxygen. C. ATPD. Water. Answer: B. Oxygen. Explanation: in the electron transport chain, oxygen is the final electron acceptor, combining with electrons and protons to form water. Conclusion: the metabolism of carbohydrates is an essential biochemical process that powers cells by breaking down glucose and storing energy in the form of ATP. Glycolysis, the citric acid cycle, oxidative phosphorylation, and glycogen metabolism are core components of this pathway, enabling organisms to convert chemical energy into usable forms. Regulation of these pathways ensures that cells adapt to changing energy demands, which is critical for maintaining cellular function and homeostasis. Let me know when you're ready for the next topic. You said lipids, metabolism. ChatGPT said metabolism of lipids. CSIR net JRF Life Sciences full length theory lecture, paragraph format, no tables, maximum detail. Previous year questions with answers in here. Lipids are a diverse group of organic molecules that are crucial for many cellular functions, including energy storage, membrane structure, and signaling. The metabolism of lipids involves both catabolic pathways, which break down lipids to release energy, and anabolic pathways, which synthesize lipids from smaller precursors. Lipid metabolism includes the digestion, absorption, transport, and degradation of fatty acids, glycerol, and cholesterol. 1. Digestion and absorption of lipids. Dietary lipids, primarily triglycerides, triacylglycerols, are broken down in the digestive tract by pancreatic lipase. The lipase hydrolyzes the ester bonds of triglycerides, releasing fatty acids and glycerol. These products are then absorbed by the small intestine, reassembled into triglycerides, and packaged into chylomicrons, lipid protein complexes that transport lipids via the lymphatic system to the bloodstream. Once in the bloodstream, chylomicrons deliver fatty acids to tissues such as muscle and adipose tissue. Fatty acids can then be stored or oxidized for energy. 2. Beta oxidation, fatty acid catabolism. Fatty acids are primarily catabolized in the mitochondria through a process known as beta oxidation, which breaks down fatty acids into acetyl-CoA units. Each round of beta oxidation removes a 2-carbon fragment from the fatty acid chain and produces NADH, FADH2, and acetyl-CoA. Steps in beta oxidation. Activation. Fatty acids are first activated to fatty acyl-CoA in the cytoplasm by acyl-CoA synthetase. Transport. Fatty acyl-CoA is then transported into the mitochondria via the Carnitine shuttle. Oxidation. In the mitochondrial matrix, the fatty acyl-CoA undergoes a 4-step process involving oxidation, hydration, oxidation, and dialysis to produce acetyl-CoA. Energy production. Each round of beta oxidation produces 1 FADH2, 1 NADH, and 1 acetyl-CoA, which enters the citric acid cycle TCA cycle for further oxidation to produce ATP. The breakdown of palmitic acid C16, 0 for example, yields 106 ATP molecules. This makes fatty acid oxidation a highly efficient way of producing energy. 3. Ketogenesis. When carbohydrate availability is low, as in fasting or prolonged exercise, the liver converts excess acetyl-CoA into ketone bodies, specifically acetoacetate, beta-hydroxybutyrate, and acetone. This process, known as ketogenesis, occurs in the liver mitochondria. Ketone bodies are released into the bloodstream, where they are taken up by tissues such as the brain and muscle for energy production. This is especially important during prolonged fasting, when glucose reserves are depleted. The process is tightly regulated by insulin and glucagon. Elevated glucagon levels promote ketogenesis, while insulin inhibits it. 4. Lipogenesis, fatty acid synthesis. The synthesis of fatty acids from acetyl-CoA is called lipogenesis, and it primarily occurs in the cytoplasm of liver and adipose tissue cells. Acetyl-CoA from the citric acid cycle is carboxylated to form malonyl-CoA by acetyl-CoA carboxylase, the rate-limiting enzyme in fatty acid synthesis. Fatty acid synthase then catalyzes the elongation of the fatty acid chain, adding 2 carbon units derived from malonyl-CoA in a series of reactions. The newly synthesized fatty acids are then esterified to glycerol to form triglycerides, which are either stored in adipose tissue or used for membrane synthesis. Regulation. Lipogenesis is regulated by insulin, which promotes fatty acid synthesis by activating acetyl-CoA carboxylase and fatty acid synthase. Conversely, glucagon and epinephrine inhibit this process. 5. Cholesterol metabolism. Cholesterol, a crucial component of cell membranes and precursor to steroid hormones, bile acids, and vitamin D, is synthesized in the liver. The mevalonate pathway is the primary pathway for cholesterol biosynthesis. Acetyl-CoA is converted into HMG-CoA by HMG-CoA reductase, the rate-limiting enzyme in cholesterol synthesis. HMG-CoA reductase is inhibited by the statin drugs, which lower cholesterol levels. Cholesterol is also obtained from the diet through chylomicrons and can be transported via lipoproteins, e.g., LDL, HDL, in the bloodstream to various tissues for membrane incorporation or bile acid synthesis. Regulation. Cholesterol synthesis is regulated by feedback inhibition. High levels of cholesterol inhibit HMG-CoA reductase, reducing cholesterol synthesis. 6. Lipoproteins and lipid transport. Lipid transport in the body is facilitated by lipoproteins, which are complexes of lipids and proteins. Lipoproteins serve to transport lipids, including cholesterol and triglycerides, through the aqueous bloodstream. Chylomicrons transport dietary triglycerides from the intestines to tissues. Very low-density lipoproteins, VLDL, and low-density lipoproteins, LDL, transport liver-synthesized triglycerides and cholesterol to peripheral tissues. High-density lipoproteins, HDL, are involved in reverse cholesterol transport, collecting excess cholesterol from peripheral tissues and returning it to the liver. Regulation. Lipoprotein metabolism is tightly controlled by enzymes such as lipoprotein lipase, LPL, which hydrolyzes triglycerides in lipoproteins, and cholesterol ester transfer protein, CETP, which transfers cholesterol between lipoproteins. 7. Fat storage and mobilization. Fat is primarily stored as triglycerides in adipocytes. When energy is needed, triglycerides are broken down by the enzyme hormone-sensitive lipase, HSL, into fatty acids and glycerol, a process called lipolysis. These fatty acids are then released into the bloodstream and transported to tissues for oxidation. 8. Regulation of lipid metabolism. Lipid metabolism is regulated by various hormones. Insulin stimulates lipogenesis and fat storage while inhibiting lipolysis. Glucagon and epinephrine promote lipolysis by activating hormone-sensitive lipase. Cortisol promotes lipogenesis and also induces the mobilization of fatty acids during stress. Previous year CSIR net questions. Question 1. CSIR net June 2017. Which of the following enzymes is responsible for the synthesis of malonyl-CoA during fatty acid synthesis? A. Acetyl-CoA carboxylase. B. Fatty acid synthase. C. HMG-CoA reductase. D. Lipoprotein lipase. Answer. Acetyl-CoA carboxylase. Explanation. Acetyl-CoA carboxylase catalyzes the conversion of acetyl-CoA to malonyl-CoA, a key step in fatty acid synthesis. Question 2. CSIR net Deck 2018. In the process of beta oxidation, which molecule is produced from each cycle? A. ATP. B. NADHC. FADH2. D. All of the above. Answer. D. All of the above. Explanation. Each cycle of beta oxidation produces one NADH, one FADH2, and one acetyl-CoA, which can enter the citric acid cycle for further ATP production. Question 3. CSIR net June 2020. Which lipoprotein is responsible for the transport of cholesterol from peripheral tissues to the liver? A. LDL. B. VLDL. C. HDL. D. Chylomicrons. Answer. C. HDL. Explanation. HDL, high-density lipoprotein, is involved in reverse cholesterol transport, carrying excess cholesterol from peripheral tissues back to the liver for excretion or recycling. Conclusion. The metabolism of lipids plays a critical role in energy storage and cellular function. Lipid catabolism, through processes such as beta oxidation and ketogenesis, provides an efficient means of energy production, especially during fasting or prolonged exercise. The synthesis and regulation of lipids, including fatty acids and cholesterol, ensure that cells have the required resources for membrane synthesis, signaling, and energy storage. Understanding lipid metabolism is crucial for comprehending how the body adapts to varying energy demands. Let me know when you're ready for the next topic. You said. Amino acids metabolism. ChatGPT said. Amino acid metabolism. CSIR net JRF Life Sciences. Full length theory lecture. Paragraph format. No tables. Maximum detail. Previous year questions with answers in here. Amino acids are the building blocks of proteins and serve as key intermediates in various metabolic pathways. The metabolism of amino acids involves their synthesis, catabolism, and conversion into other bioactive molecules like neurotransmitters, hormones, and nucleotides. Amino acid metabolism is a highly regulated process that helps maintain nitrogen balance, support protein synthesis, and provide precursors for energy production and biosynthesis. 1. Amino acid catabolism. Amino acid catabolism involves the breakdown of amino acids into simpler molecules that can be used in various metabolic processes. The first step in amino acid catabolism is the removal of the amino group, a process called deamination. The amino group is transferred to alpha-ketoglutarate to form glutamate via transamination, catalyzed by aminotransferase enzymes. Glutamate can then undergo oxidative deamination by glutamate dehydrogenase to release ammonia NH, which is subsequently converted into urea via the urea cycle for excretion through urine. The remaining carbon skeleton, now in the form of alpha-keto acids, can be converted into intermediates of the citric acid cycle, e.g., acetyl-CoA, pyruvate, oxaloacetate, where they can be used for energy production. 2. Urea cycle, ornithine cycle. The urea cycle is a key metabolic pathway for the removal of excess nitrogen, primarily from amino acid catabolism. The ammonia produced by oxidative deamination is toxic, so it must be converted to less toxic substance, urea, in the liver. The urea cycle involves the following steps. Ammonia reacts with carbon dioxide to form carbamoyl phosphate, which then enters the cycle. Citrulline is synthesized from carbamoyl phosphate and ornithine. Arginine-O-6-onate is formed from citrulline and aspartate. The final step involves the conversion of arginine into urea and ornithine, the latter of which can re-enter the cycle. Urea is then transported to the kidneys for excretion. The regulation of the urea cycle ensures that nitrogen is efficiently eliminated from the body without accumulating toxic levels of ammonia. 3. Glucogenic and ketogenic amino acids. Amino acids are classified as either glucogenic, ketogenic, or both based on the metabolic fate of their carbon skeletons. Glucogenic amino acids are those whose carbon skeletons can be converted into glucose through gluconeogenesis. Examples include alanine, glutamine, serine, and glycine. Ketogenic amino acids are those whose carbon skeletons are converted into ketone bodies, e.g., acetyl-CoA, acetoacetate, which can serve as an alternative energy source during periods of fasting. Examples include leucine and lysine. Some amino acids, like phenylalanine and tyrosine, are both glucogenic and ketogenic because their catabolism produces intermediates that can enter both the gluconeogenesis and ketogenesis pathways. 4. Amino acid biosynthesis. The biosynthesis of amino acids refers to the synthesis of non-essential amino acids, which the body can produce from precursors. Essential amino acids, on the other hand, must be obtained from the diet as the body cannot synthesize them. The synthesis of amino acids typically involves the following key intermediates. Alpha-ketoglutarate, a precursor for the synthesis of glutamate and other amino acids like glutamine, proline, and arginine. 3-phosphoglycerate, a precursor for the synthesis of serine, which can be converted into glycine and cysteine. Aspartate, a precursor for the synthesis of asparagine and other amino acids like methionine, threonine, and lysine. Pyruvate, a precursor for the synthesis of alanine and other amino acids like valine, leucine, and isoleucine. The biosynthesis of non-essential amino acids is often regulated by feedback inhibition, where the end product inhibits the activity of the enzymes involved in its synthesis. 5. Amino acids as precursors for other bioactive molecules. Amino acids are not only used to build proteins but also serve as precursors for various bioactive molecules. Neurotransmitters. Amino acids like glutamate, glycine, and GABA gamma aminobutyric acid are critical neurotransmitters in the nervous system. Hormones. Amino acids like tryptophan are precursors for serotonin, and tyrosine is a precursor for dopamine, epinephrine, and norepinephrine. Purines and pyrimidines. Amino acids like glutamine, glycine, and aspartate contribute to the biosynthesis of purine and pyrimidine nucleotides, which are essential for DNA and RNA synthesis. 6. Disorders related to amino acid metabolism. Several metabolic disorders arise from defects in amino acid metabolism. These disorders can result in the accumulation of toxic intermediates or the inability to synthesize essential molecules. Phenylketonuria (PKU), a deficiency in phenylalanine hydroxylase, which leads to the accumulation of phenylalanine and its toxic metabolites. Maple syrup urine disease, a defect in the catabolism of branched-chain amino acids, e.g., valine, leucine, and isoleucine, leading to their accumulation in the blood and urine. Alkaptonuria, a deficiency in homogentic oxidase, leading to the accumulation of homogenic acid and darkening of the urine. 7. Regulation of amino acid metabolism. The metabolism of amino acids is tightly regulated by several mechanisms. Allosteric regulation. Many enzymes involved in amino acid metabolism are regulated by feedback inhibition from the end products of the pathways they control. Hormonal regulation. Insulin and glucagon play key roles in regulating amino acid metabolism, particularly in relation to protein synthesis and breakdown. Insulin promotes protein synthesis and amino acid uptake into cells, while glucagon and cortisol promote protein degradation and amino acid release. Previous year CSIR net questions. Question 1. CSIR net June 2018. Which of the following amino acids is exclusively ketogenic? A. Leucine. B. Glutamine. C. Alanine. D.